Having considered the vastly different members of the lipid family, let's see which ones contribute to the formation of biological membranes. Every living cell has a cell membrane. This includes single-celled microbes as well as multicellular plants and animals. You'll want to call to mind the difference here between prokaryotes and eukaryotes. For though both have cellular membranes, eukaryotes also have membrane-enclosed organelles, such as the nucleus and mitochondrion. A membrane always presents an effective barrier to separate compartments and set up two different environments. If we consider just the lipid portion of membranes, the driving force of their associations is the hydrophobic effect. As a matter of fact, though, membranes are composed of both lipids and protein. Each of these components relates to the structure and function of membranes. Since these membranes are effective barriers, this necessitates a means of transporting substances across this barrier. Membranes also serve another function in that within them is housed important enzymes that could not function outside of that environment. The lipid bilayer is an aggregation of lipid molecules that associate in such a way as to minimize the ordering of water molecules. The bilayer consists of two monolayers of lipids with their polar head groups associated and facing the aqueous environment within and outside the cell. Recall that the lipid head groups always carry charges and can therefore interact strongly with the polar water molecules. From the figure below, note also the position of the hydrophobic tails. They are facing the internal region in association with each other and in exclusion from the water environment. Also remember that these hydrophobic tails are fatty acid chains of variable lengths and degrees of saturation. A small volume of lipids may combine to form a vesicle, as illustrated below. Note the presence of the lipid bilayer and the inner aqueous compartment. Due to the geometry of this arrangement, the inner layer of lipids is more tightly packed. The lipid bilayer is therefore held together through multiple van der Waals interactions. Not only do the lipid tails vary, but also the nature of the head groups. This means a mixture of lipids within the bilayer, with bulkier molecules occupying the larger volume of the outer layer, and smaller lipids within the inner layer. This last point may be more apparent by examining the prevalence of different lipids in the composition of rat liver cell membranes. Look at the last column of the table given here to see the variety of lipids seen in the plasma membrane of these cells. Phosphatidylcholine is present at twice the concentration of sphingolipids, but equal to the concentration of cholesterol. Now notice the first row to see the prevalence of phosphatidylcholine in different membrane structures. For instance, the highest concentration of this lipid is found in the nuclear membrane. It's not important that you memorize these values or even the specific trends. Rather, simply realize that lipid composition varies between membranes, and this very much relates to their functions within these structures.